Welcome to Taxonomy Tuesday, everybody. Be your host this evening. If anybody's got anything to share from their computer, want to start us off while I check for some emails, that would be awesome.
I have stuff to show, but I'm not quite ready. So maybe I'm going to do this a little bit later.
Chris, I can try to share from my phone while you're getting emails together.
Go for it.
All right.
There we go. I don't know if you're talking as well, but you're on mute.
Sorry about that. Yes, I was talking. So you can hear me, right?
Yeah.
Okay, cool. Yeah, I don't know what this is. It's a myxomycete. It's a slime mold. And honestly, I tried keying it out, but I can't even get it to an order. I don't think it's a metatrichia. You can kind of tell that it's a bunch of sporocarbs coming off of one stalk. And you can see the stalk better in this photo. And that stalk has like a couple feet, right? It's kind of branched at the bottom, and then the sporocarbs.
It really spread out.
Yeah. It looks like there's maybe at least two, maybe three, maybe a third kind of spoke foot going off in the back there. And you can see they got a little black tip to them. This photo is not great. But yeah, I figured I'd share that one. I'm not quite, I really don't know which one it is. I tagged a couple experts, so hopefully someone can help me out.
Could it be Arcryia, cinea?
So, yes, it could be Arcryia cinereum. But as far as I can tell, that does have it sometimes, because those have one head to one stalk, you know. But sometimes those stalks can kind of grow together and give the illusion that there's only one stalk and that it's a little cluster thing going on. I think. Maybe I could be wrong on that. And Arcryia cinerea can present as having, multiple uh, fertile structures on the sterile foot. But
Yeah,
I
it almost looks like it opened.
that's, I guess, kind of another possibility. Yeah, I don't know.
It's a
Because I
winner.
think, yeah, things like, metatrichia, so a lot of things in tricky alleys, which Arcryia cinerea, Arcryia is in tricky alleys, the order. Um, like Metatrichia, for example, has several kind of sporocarps on one stalk. So I was looking in that direction, but nothing really fit there. So yeah, not sure.
I put a picture of what we hear labeled Arcryia last year that I had found in the chat. And I would think we decided that the legs had fused together, like you are saying. Um,
Yeah.
And do you have any other pictures of this? Is this in situ or what's going on? This is under oh yeah, that's right.
Yeah. And it's not a great photo, so it's not very helpful. I mean, you can kind of get an overall picture, but they're all that way, is I think what you can gather from this one.
Are they dried up?
Not so much.
Okay. Yeah, so that looks really different than mine. Okay.
Is the black part a hole?
No, no. When
Hmm.
I'm zooming, when I zoom, can you see the zoom?
Yeah.
Yeah.
Okay. Yeah, no, it's not a hole. It's they all seemingly have like just black tips, even in this very blurry out of focus one. You can
Oh,
still see it.
really, you can see it there. Yeah. Hmm.
And this one shows this photo, the first one kind of you can see the head while the back. I need to learn how to photo stack. Otherwise,
Yes.
but yeah, you can kind of see the back row of sporocarps pretty well. That's in focus here. And then this last one, the foot was kind of what I was interested in most because, Hunter, like you were saying, is this an Arcryia cinerea where the foot, the stems, the stalks are fused together? And I don't think it is, but it could be. So it's not very helpful.
At the base,
You could
it looks kind of like there's two fused together or a branch or I don't know.
Yeah, like branches on the feet almost.
Yeah.
could you scroll over to the one that's on the bottom leaned over? Because that one is in focus.
This one?
Oh yeah. Yeah, now that doesn't look like it opened as much as it's separate, I don't know, raisin looking things.
Yeah, sporocarps. And that's the other part.
It doesn't look like you could just blow these out, you know?
It's not at that level of maturity. See this one in the back, way at the top there?
Fuzzy top.
Fuzzy top. So that one I could have gotten some useful microscopy out of. But I actually didn't notice that until I was just sharing this photo. But I under the scope, I didn't find much information just because, the peridium on each of these sporocarps is still closed. Hmm.
Did INAT say anything?
Yeah, but I mean, they suggested Arcryia cinerea, but
Oh, okay. Mm-hmm.
um,
But you don't,
I,
you think there's, something else.
I think
Yeah.
it's something else. But I'm not 100% sure it's not that. And also, you know, uh, I was looking at a Cribraria. I don't think I shared it here a few weeks ago, and I just couldn't figure out this Cribraria. It looks like cancellata, but it has a calyculus. So I tagged someone on INAT, and they gave me a variety of Cribraria cancellata that actually has a calyculus. So I was like racking my brain. I was like, it can't be cancellata because it has a calyculus, and then they ID'd it as a variety that
Could you
actually
remind us what a
does.
calyculus is?
Sure. Let me go on to the next thing. And this photo came out real good.
Oh, wow. That's amazing.
Yeah, this might be one of the better photos I've ever taken. Um, so the calyculus is this goblet-looking part, that membranous-looking part. The cone to the ice cream, if you will.
Okay.
Oh,
Uh,
okay. Mm-hmm.
that membranous bit where the capillicium is kind of coming out of.
Oh, wow. Nice.
Now, did these like mature under like your possession?
No, No, I found these. I looked at these the same day I collected them. So that was the stage of maturity that they were in situ. But yeah, this is Hemitrichia. Oh, and I think on the last one, when I said Hemitrichia, I meant to say Metatrichia, not Hemitrichia. This is actually Hemitrichia.
I think previously you said Metatrichia.
Oh, I did. Okay,
Yes,
cool.
and I remember it because I don't know what that is.
The wasp nest slime mold.
That's Metatrichia. Oh, okay.
Yep.
Oh, I do know what that is. Then I just forgot
Yep.
the name. Yeah.
Metatrichia vesparia.
Okay.
Yeah. And the way to work out the species with these Hemitrichia, there's two species that this could realistically be, and they are very hard to work out. The one that I chose, which I think is calyculata, I'll go back to the name here in a second. And this one, now that I'm looking at it, kind of does look like it has a halo. This species is supposed to not have the halo. Whatever the other species is does have an obvious halo, but calyculata also has this kind of odd inclusion, kind of that irregular. And I'm looking at the spore between 4 and 3.5 integer there, just to the left of that. But yeah, more or less the spore kind of, in my mind, supported calyculata more, but they are pretty difficult to work out, the two species.
Really cool.
Yeah, thanks. Any other questions on this one? I think this might be my last slime mold that I wanted to share.
That's a good photo contest one, though, right there.
Yeah, I was proud of that one for sure. Yeah, this thing. So this was from the foray as well. And this is a species of Thelephora, presumably. I think I have a good handle on that at least.
Yeah, it's a Thelephora.
Yeah. Palmata or whatever I chose here. Yeah, palmata.
What does your description say it smelled like? The bay?
Yeah, the bay.
Oh, okay.
Yeah, it did not spell good at all. I mean, yeah. Yeah, it smelled like
not in a good way. Okay.
Right. But I will say, for this identification, this is supposed to be a conifer associate. And I'm not sure if I saw, if there were any conifers in the area. I missed it. I don't think there were any conifers there. So maybe this is a different species.
It's pretty cool looking. It's different from the ones I've seen. Really neat. Not of this species, but the genus. That's cool. Yeah. Neat.
Are the little tips on the end like a feature?
So I think the description for this one says it starts off with it can be kind of rounded or tipped, but in maturity these things kind of fan out. I that's what I was reading at least. So, but the odor, it's supposed to stink. This one, what's the common name? Stinking earth fan, and Thelephora in general, most of them smell pretty bad. So, for this one to be the stinkiest of all the Thelephora is saying something.
That's awesome. I'm glad you showed that. I was wondering which species, what it looked like. The really smell, the one with the name, the stinking. Yeah,
Cool.
yeah.
Yeah, and
Cool.
Dave, you said you thought this was a Thelephora as well, and I think so. Do you have any opinion on the species here?
I was just looking on Michael Quebec now, but I think there might only be one that looks like this. There's quite a bit of um, visual diversity, morphological diversity in Thelephora. I'm not sure how many there are that have these like lots of little projections, lots of long projections sort of fused together. I'm going to look. I'll look now.
Yeah, the other species, and they have it on there that I was considering, started with an A. It was a really long name. Yeah, I forget what it was, but it started with an A. So while you're looking, check that one out as well.
So was that flexible, but hardy?
Yeah, yes. So
Mm-hmm.
that's a good question, because when I saw it, and I'd never seen, I don't think I've ever found this particular, whatever this thing is, I don't think I've ever found it before, but it definitely had that same Thelephora feel to it.
Yeah.
Like when you find Thelephora, what is it? Oh, terrestris, right? Which is the common one in the pine barrens. It's got, yeah, it's kind of rubbery. It kind of would be, it's not as fragile as like a clavaria or something like that, if that makes sense.
Yeah. It's almost like a fan, like a brush or a fan, or a little bit.
What, Brendan? What's the species name you used here?
Palmata.
Palmata. Palmata. Okay, let's see. Oh, they have it here. Yeah. That looks like it. And the other one is the one that starts with A is let me see. I just had it. Oh, Anthocephala. Anthocephala.
Okay. Yeah.
Yeah. That one looks pretty similar, actually. Yeah, I'm
Yeah.
not sure how I would tell these apart in the field. Let's see if there's any kind of difference in spores.
I think there was. This one's on my, I have a backlog on microscopy.
Yeah.
But this one I definitely have pegged for microscopy, because I think they have interesting spores for sure. Um, this name, the palmata, they're like attenuate, or like really, really hairy and kind of oddly shaped, almost like hairy anosope spores.
Yeah, attenuate. They're saying on Michael Quebec. And I think that basically
Thanks.
means what you're saying. Um, angular to angular lobed. Oh, there's a picture here
Yeah, the
of the
picture is really
Palmata
interesting.
spores. Yeah.
Yeah.
Yeah, I've looked at these things before. Yeah, they're really weird looking. They're hard to make sense out of.
Yeah, and what range? Because some of them look more like almost like a turkey tail or something. Some of the species, and
Yeah.
then the others are more like brush-like or
Yeah.
fan.
Yeah, that's what I was saying. There's a lot of diversity in the way they look.
Yeah, really neat.
Nice.
It sounds
To
like
keep
these two species under consideration, Anthocephala and Palmata, have spores that are not that much different from the descriptions. Let's look at Anthocephala. What does it smell like?
Yeah, because that was the other thing that kind of gave me some pause. I mean, the Conifer Association really, really should put a halt on this name. Um, but the other thing was the odor. Me and Nicole both notched it as oh, it's kind of fishy, like in a bad way. And as soon as I said the bay at low tide, yep, that's exactly what it smells like. And I think the palmata is supposed to smell like, what is it, rotten garlic? Which
It
those two,
smells bad. I've found it. It
yeah,
smells bad. It's like garbage, sort of rotten stuff.
Yum.
Yeah, it says about Anthocephala, indistinct smell and taste.
right.
Well, I wouldn't want to taste these things.
I'm right there with you.
Well, it's a brand new rabbit hole.
All right. Don't want to take up too much time, but I did mention that I found some earth tongues. This was not from the foray, but this is Trichoglossum walterii, or walteri. They're small. Have that kind of spoon shape to them, that kind of concave. It's tough to make out in a photo, but I think you can kind of see it with the shadow there.
Oh yeah. Oh wow, cool. Neat.
Yeah, so I love working with these things under the microscope. Um, so what we're looking at here, all those black pointy things, they're actually, uh, what is it, setae? Little hairs on the outside of Trichoglossum, and that's, I've shared these before. So Trichoglossum is the one with the hairs, Geoglossum is the one without the hairs. So you find the hairs, you know what genus you're in at least. Um, and then with these, you got to count the septa. So how many of those segments there are. In this case, there are 7 pretty much across the board. That's the end of the hairs. And you can see they are pretty pointed. Some species describe the end of the hairs and the color of the hairs differently. These are pretty dark. That's an ascus. You can see it starts up here at the 10, runs all the way down to the 2 or so. And this is probably, well, let's see, 204. Yeah, so that's at 400.
So I was just having a technical issue. So I'm just coming back. Those are setae, those pointed things?
Yes.
Yes. Okay, good. Because I know I got confused once before by it was setae confused with immature asci, maybe? I'm not sure. I confused them with something.
Yeah, the immature ones, depending on what species you're looking at, they can be pretty skinny and you can definitely confuse those.
I think that's what I did once. Okay, so they're setae. So Trichoglossum, those are the ones with the setae, I guess. I think, yeah.
Yeah. Yep.
Yeah. And then
And
like you
then,
said, you just count the septa on the spores. They're usually pretty consistent. And there's a couple of species where they vary from like 13 to 15 or something, but most of them have like the same number of setae all the time. Or not setae,
yep.
septa. 2, 3, 4, 5. This is the one with 7. All right.
Yeah, and even the ones that are, 13 to 17 or whatever it is,
Yeah.
that's also pretty consistent, So
Yes, yes. That's what I have found with these when I find them. I don't find them all that often.
And then the other thing with these, if you want to get to a species, you got to check out the paraphyses, which are the sterile structures under the microscope. They're infertile. And you can see these are kind of a little bit all over the place, which kind of fits the bill for the species, Walterii. They can be crescent, they can be hooked. Here we're seeing a bunch of them that are between crescent and hooked. Some of them, like this one up top here, a little bit of a subglobose thing going on.
What about this? Is this one right here in the lower left? It's got several turns in it.
Yeah.
Yeah.
Right. So that's kind of that all over the place portion of this. And you also want to see, you can see that the paraphyses they're also septate. So they have segmentations as well, which you can see right there. Also right there. Pretty much all over this photo. And you want to see if there is a constriction at those septations or if it's more relatively straight. I think there's a good one where you can see it's kind of out of focus, but you can make out that it's constricted at those segments. And then this one's almost cane shaped. So all that fits walterii. And this is where I found it. This is where I most often find my my earth tongues. This is next to a stream, but I love looking for them in mossy areas next to bodies of water. That's where they are down there.
Oh yeah, the last ones I found, that's exactly the habitat. Exactly.
Mm-hmm. Yeah, they love that, and you know they love mosses of all kind, different species. Like maybe prefer sphagnum. Different others might prefer this type of moss, but they love sphagnum too.
Nice.
Habitat shot.
Yeah, I need to be doing that. I need to be doing that more often. Just to kind of give us an idea of where these things are found. You know.
It's a good.
Yeah.
But cool. I think that'll do it for me. Yeah.
I have a few. If you want, I can share before going to emails, Chris. If you want.
Go ahead.
Okay.
Okay. I've stopped sharing officially, right?
Yep.
Okay, cool. Thanks, guys.
Thank you.
Can you see iNet?
Yeah.
Oh,
Yes.
good. Okay, very good. So I don't have a lot of great stuff, but this was a really cool color mushroom in the woods this morning, and I almost thought it was uh odoro, but what is it? Colostomy? What am I thinking? Clitostomy?
Mycosby odora.
Yeah,
Right.
but that's blue, and this was clearly green. And I mean, I think it's just another Russula. But it was really, really pretty. And I put some KOH on it and got this reaction. And I did the cuticle peel, and it was about 100%. It was real. It wasn't very dry. It was rubbery. It was more like a Suillus, almost like a Suillus cuticle. It was kind of interesting. And then all I can show spore wise is this. I'm getting a new phone. I'm done with my phone, but I just couldn't get it to focus, and it was dirty. And at any rate, they were heavily ornamented spores, um, and pretty cool spores. Sorry, I can't show you a better picture, but I thought it was a really pretty Russula, and I think it could be. I mean, just very agata, but probably not. I don't know. Anyone have any thoughts? I didn't take it.
I don't think it's variata. I didn't see any forking of the gills near the margin of the cap.
Okay. I just didn't have the time to do anything
I don't.
more.
Let's look carefully. Is there any forking? I don't see any forking.
I don't really
It's a little
either.
hard to tell, but I can't see forking.
No.
Yeah, Variata would have a lot of forking
Okay.
at different distances. That's why it's called Variata, actually, the variably spaced forks.
Oh, okay.
That's what I've heard. I don't know. The color is also very variable.
Variable.
Sometimes it's all green,
Sometimes it's purple too, isn't it?
purple, green, black, mottled.
Yeah.
It's a lot of different colors.
Yeah, and this had the dark in the center that you'll see on some Russulas. So I need to look around and see if I can figure out what it was, but I liked finding it. It was very pretty. Um, so there's that, and then we've seen this before, not that long ago, but this was on one of my shiitake logs, in the shiitake space, whatever, in the backyard. And it was cool. I growing right on one of the logs, and you can see them in every stage here, which is really neat. I'm pretty sure it's Crucibulum laeve. I have a picture of whoops. Um, uh, where's what? Oh, here. So here's the side view of the nests. And then I tried to do some microscopy, and that was interesting. So they were really hard to crush, the eggs. They just really wouldn't crush. I just smashed them down, and I really couldn't. I thought I'd be able to make a fine dust out of them, but I couldn't. And so this is one of those squished eggs, and then I believe this material came out from the inside, and it was. But I might be wrong on that. But it looked like this, and it seems to maybe have. I don't know if those are clamp connections or spores, like those the little well, you can see, I think, what I'm blowing up just to the right of the eye. Um, there's a few things like that, or this these bulges. I don't know if those. I don't know what those are, but maybe someone else does. Um, and so that was it. It was a nice find, and I've got a couple other just pictures of some finds I can show you
Hey,
guys.
Hunter,
Yeah.
can you go
You
back
know
to
what?
the
I think I don't remember. I haven't studied these birds' nests for a while, but I think the spores actually form on the surface of the eggs.
Oh,
And I
okay.
think you can just put some eggs on a slide and let them drop off. I think that's all I did. Let me see
Oh,
what Michael Quebec says. I don't think you have to smash them apart.
okay.
well
Maybe just
that
even scraping them a little would be better.
Yeah, I think
I
well,
was wondering if you could go back to the smashed peridial micro.
Sure. Like this one or this one.
I missed the photo.
Oh,
But you
I'm sorry.
said you smashed one of the peridials? Yeah.
Yeah, yeah. I smashed a bunch of them, but um, yeah. This is what you wanted to see. Can you see it?
Yeah. Yeah,
Oh,
yeah, I can.
okay. Yeah, and I
Because I
mean,
was under the impression, and I'll be honest, I think I've only ever done the micro on these once and ever kind of really tried to get into it one time. Um, but I think the micro that I did when I smashed the peridial, I cut it in half, or like I punctured it and then did a smash mount. Um,
mm,
and I thought the spores were coming from the inside of the thing, but I could be wrong.
I thought the spores are supposed to be inside, But I mean, at some point, the raindrops splash these out and they break open or something. And I mean, the first, well, I mean, I thought I took like one of the most mature ones, but I mean, a raindrop would not have broken, splash. It wouldn't have like come open, it was really, so I don't know. Even cutting it in half seems like it would have been difficult, but
Yeah, because I remember cutting one in half, and it was like cutting
walnut.
like cutting a seed, a little
Yeah,
tiny seed or something.
right. Yeah, it was really hard. Yeah, exactly. Okay, well, I agree that I thought they were inside, and but we could be wrong, but I agree, and that's why I was trying to like just smash it, up, break it up, smash it up, and that just wasn't that easy. But so probably there aren't spores in here, and this is, you called it what? This
Found I
material.
found some stuff here.
What'd you
Uh,
find, Chris?
Under a microscope, birds' nest fungi reveal fascinating reproductive structures, including large, smooth basidiospores, complex, multi-layered peridial walls, and microscopic coiled anchoring threads called funicular cords.
I think that's what these are.
That's what I'm thinking.
Yeah, and I think the spores like spring out off of these or something
Let's see what else we got
when the
here.
rain hits them.
Basidiospores, large, oval to broadly elliptical, smooth, high-line, and thick-walled. Peridial, peridiolum cortex, the outer shell of the egg, contains dark melanized pigment and hyphal walls protecting the internal spores.
Yeah, it looks like I was wrong. Michael Quebec says that peridial disintegrates and then the spores are released.
Acidia,
So they're
club-shaped
inside.
spore-bearing cells visible inside immature peridials.
Okay, so can you describe what did you call these again? Coiled. I'm just
Funicular cords.
coiled funicular cords. Cool. Cool. Good.
It's a complex structure of interwoven, tightly coiled hyphae that anchors the peridial to the inside of the splash cup, which unfurls and sticks when wet.
The splash cup. Yeah, right. That's neat. So yeah. Okay. Yeah, cool. All right.
I just put not this species, but I put a link to an observation that I had where you can see the peridial that I was talking about that I cut in half, and you can see the spores kind of coming out for whoever wants to take a look at it.
Oh, well, Chris, can you open it? You want to open it for everyone? Or am I
Yeah.
doing that right now?
You might be doing that right now.
Okay, cool.
Oh, nice.
Yeah, perfect. Okay. And
Yeah.
so yeah, this is like a different one, but I think, right? Like
Yeah.
yeah,
Yeah, but
maybe.
if you want to go to whatever micro shot it is, you can kind of see the first one, I think.
This?
No, the one before this.
Oh, this?
Yes. Yeah. So you can see that, melaninized outer portion. Uh,
Okay,
and
so
then
I saw
all
stuff
this,
like this under the microscope, but I didn't know what to do with it. I didn't know what it meant. What is this?
well, it's just a bunch of spores coming out of the inside of the egg.
So those are the spores like right here in the middle?
Mm, yep.
Yeah, okay. So I did see that too, but I just didn't know if they were them. And like they were hyaline, more so than these. So
Well, this is probably at my 100x, which is why, the detail isn't all that great. But you can also see those strands at the top there.
these
Oh, off to the right, too. Yeah.
Yeah.
right, and this species they might not be as high a line either.
Sure.
Yeah, neat. Oh, that's cool. Neat. Okay, good. Yeah, I wasn't sure if I was looking at the spores or not. So, okay. Good, good.
Yeah, that one looks like it's got a clamp
right.
connection on it.
Yeah.
Right here?
Yep.
Yep.
Yep. Nice. Cool. Okay,
Yeah,
good.
I just wanted to share that cross section of the peridial, which is the egg.
Yeah, I see.
And I'm glad we got the supporting information there, Chris. So
Yeah, this is very good. Very good. Okay, cool. All right. And then I just have just a pick or two just a pick or two really quick. I found my first scleroderma of the year. And I'm surprised I haven't found more yet. And I had wanted to do microscopy, but I went down there. I thought I put it on a slide, and it just wasn't there. And then I found a beautiful fulva, I believe, but I didn't pick this at all. But this was really gorgeous. The color is true. Um, very pretty. And then the train wrecker.
What did you call the Amanita? Did you give it a species?
Fulva.
Fulva, yeah, that's what I would say.
Okay.
Yeah, it's good to see the vulva though, like you said. See if it's white membranous vulva with stains.
Sure. And then lastly, I don't think this is fungus at all, but when I first saw it in the moss, I thought I found Matrula in this really wet, mossy area. And I was like, Matrula, because I was up in Wisconsin and I was so excited. But then I started kind of tearing apart the moss, and this is, I don't think it's a fungus at all, but can someone tell me about it? Is it a flower of the moss, or does anyone know?
Was it on a
mean,
stick? Is it on a stick?
Kind of, yeah. This was very, stick-like, right. It's at the head of a stick-like structure inside the moss. And there was a couple of them. It wasn't the only one. But I was disappointed. It clearly isn't Matrula.
No,
No.
but it's interesting, whatever it is.
It reminded me of the soldiers, the red lichen.
Yeah, I just showed the photo to Karen. She said she doesn't think it's a lichen.
Okay.
Yeah, because I thought that also.
Okay, yeah.
Yeah, there's another lichen that forms little cap type things like that.
Right, but the red, right?
Diabases. Yeah. Yeah.
That's the soldier one.
No, that's Cladonia. Soldier's Cladonia, right?
British soldiers.
Yeah. Cladonia. That's British soldiers.
Oh.
But there's another one.
Diabeus Phaeomyces.
Diabeus Phaeomyces. Yeah,
Oh.
that's the one that's got a little pink thing on top that looks like this.
Oh, I want to check that out. Okay. All right. Well, that's it for me. And
Can you go back to the in situ photo? Was there one?
Well,
Or yeah, well, where you collected it.
yeah, this is the best I can do there.
Yeah.
Yeah.
Looks like that's probably how it looked where it was.
Mm-hmm. Exactly.
I don't know.
Yeah, okay. I will
I wouldn't rule out fungus personally.
Yeah,
Yeah, I mean, could like a tiny little stick be rotting away in the moss and like a tiny little jelly on there?
Yeah. Or, it could certainly be a myxoplasmodium.
Well, I definitely want to find out. It is very cool. Okay. Well, thanks. I'll consider those things and see what I can find out. Thanks. I'm
Thank you.
stopping the share. I think I stopped now.
Nice. Does anybody else have anything from their machine, or we move on to the mail? We have stuff from Doug and Dave and Shihong. Shihong sent her stuff in all the way on Sunday. You want to go?
Sure. Can you hear me?
Yeah. Yeah, let
Okay.
me
Can you share your
start
screen?
sharing the screen here.
Okay, so this one, so this is Sunday. I saw this in Autumn Hill Preserve. It's really just 5 minutes away from downtown Princeton. It has teeth on the bottom, and the cap looks very interesting, like feathers back, but under it's teeth. So AI gave me the name for Shinko hedgehog. Anybody have any idea? You ever saw this before?
Well, there's maybe another one or two that look a little bit like this, but I think that's probably the right idea. Did you taste it? I think they're usually bitter.
I look up. They said there are two types. One's bitter, one's edible. I don't know which is which. Yeah.
I think the edible one is collected in the Rockies,
Mhm.
where they call it hawkswing. The ones that I found here in Pennsylvania were bitter.
Okay, I did not try. It just looks
Yeah.
very unique.
You would think there would be more than one species, really.
Mhm.
I mean, given the differences in location and taste.
There's some greening that shows up on the bitter one and the stem, or when you cook it, or something. That looks green at the base, doesn't it?
I did not see green on the base. Maybe this is just the photo.
Maybe a tiny, tiny bit in the upper part of the picture, which is the base of the stalk, looking to the left. Maybe a tiny, tiny bit of greenish. You have to kind of use your imagination. It's just a little area.
I did not notice that in situ.
Yeah. This is I'm just probably seeing something like a shadow or something.
Nice.
Yeah, they're very unique looking.
Yeah, I love finding those. It's rare that I do, but that's a cool find.
Oh, wow.
Ooh.
So this is the second time I see indigo, and I saw a bunch of them. I think I have at least 6 fresh one and 3 or 4 dry one.
Lucky.
Oh, beautiful.
Yeah, blue latex.
Yeah, it's like it stained my hands blue.
Yeah.
It almost looks fictitious.
What's the word? Fictitious?
Yeah, just fake. Like, how could it be that colored? It's ridiculous.
Yeah, when you slice it, the color is even brighter.
Did you eat them?
I tried cooked one, but I was trying to cook with garlic. I did not find garlic, so I mixed it with onion. So it tastes a little bit sweet. So I don't know if it's old taste or from onion.
Nice. Where was this around?
Same place.
Oh, cool.
It's really just 5 minutes away from Princeton. It's called Autumn Hill Preserve.
So
I just took picture with iNet, so it should have the map.
Yeah. Never seen one of those before.
I was surprised. I see a bunch of them.
It looks like it was growing with oak, basically. Oak.
yeah, they say oak and pine mix.
Okay.
Is good for this, but probably either oak or pine.
Yeah, so nearby, it's when I just walk in, it's very dry. I did not expect to see anything. I start to see all sorts of Amanita. I was taking picture of Amanita, then I saw something blue, and it turned out it has a bunch of them.
That is so exciting.
Yeah, it's really a good surprise.
Yes.
So this is nearby. There are a lot more of this type of milk cap. I picked the name, but I wasn't really sure if what I have is correct.
What do you call?
I would have to check, but I think peckii is really acrid, like peppery acrid.
Okay,
So,
I did not taste that. I
yeah,
didn't even try.
tasting Lactarius and Russulaceae in general is usually helpful. I'm going to look.
Oh, I forgot to mention I tasted my little green Russula, and it was mild and unremarkable, and the smell was maybe a little sweet, and that's it. Did you get much milk on this, or was too?
This one, no, I did not even try to slice it, but when I touch it, no milk come out actually.
It looks like the one edge is pretty dried up.
Yeah. They're fresh one. I did not even bother to flip them over.
Yeah, peckii's taste is quickly burning acrid.
Okay, so I really have to.
If you taste it, you might be able to tell. Because
okay,
these Lactarius mushrooms, especially when they get old, they start to all look the same. Well, a lot of them, at least. I mean, some are distinctive, but could be.
Next time I taste it, so I will know for sure.
Yeah, none of them are really a risk to taste as long as you spit it out.
So this mushroom I see quite often. AI gave me the name. I wasn't really sure. Clitopilus.
Clitopilus prunulus.
Okay.
Yeah, did you smell it?
I smell it. I cannot really smell much. I'm never sensitive to smell. So
Yeah, they're usually you can smell this like cucumber smell, or some people call it bread dough. I
okay.
think there's more than one species in this prunulus complex
Yeah.
because I seem to find ones that look a lot different. Like the gills will be widely spaced on one, the gills are really close on another one, and they all have this cucumber odor. And the way to really tell that you have Clitopilus is well, first of all, if you get a spore print, it should be pink, almost like an entoloma print. I
Okay.
think they're actually related to entelomas. I'm not
Mm-hmm.
sure, even though these are Quadripilus prenulus is edible, at least. Well, yeah, it's I guess even if there's more than one species, they're probably all edible. But under the microscope, the spores are rather distinctive. They're shaped like footballs, and they have pointed on each end. They said the ends are tapered, and they have these ridges that run like longitudinally from one pole to the next. And
Mm-hmm.
usually, even at 400 times magnification, a lot of times you'll be able to make out one ridge, maybe on one of the spores, or maybe two ridges. Especially if you're able to, if you have a good microscope, or if you can view at 1,000, you're going to be able to see the ridges in the spores.
So it's commonly called the Miller, and you've eaten it, Dave?
Yeah,
Or
it's pretty good. Yeah,
right?
I mean, I don't, I'm not regularly. I mean, usually when I find them, I find like two,
Uh-huh.
like two or three, so I don't bother. I just take them home and get the spores and look at the spores.
Mm.
But yeah, when I've eaten them, I thought they were pretty good. You have to watch out though. You have to make sure you don't have an entoloma or a clitosoby, or there's a few things that could cause a problem. Is this
Yeah.
where are these photos from?
This is just an iNet.
Okay, yeah, there's going to be photos of the spores. If you can enlarge one of those photos, you'll see what I mean. See where you can see like a slit going. That's one of the ridge. That's one of the ridges. There's look at the one that's right in the lower middle. You can actually see two of the ridges.
Mm.
So that's a pretty good photo there. So shaped kind of like footballs or beech nuts actually, kind of, and tapered at each end to a point, and with these longitudinal ridges.
Mm.
Oh, this is good. Yeah. And you said what color spore print?
Pink.
Pink. Oh, okay. Yeah.
Yeah, kind of like an entoloma print almost.
Mm-hmm.
But that one had a.
Is there a flavor in the woods when you taste it? Did you say you said it smelled like cucumber or
I usually don't taste these. I just smell them and then I say, "Oh, that's it." And then I take them home and say, "I don't know. I better make sure." So I get some spores,
Uh-huh.
and I pretty much I've always been right about what they are, but they're
Mm.
kind of unnerving because they don't all always look the same. I think there's probably more than one species.
Yeah, if you look at the between gill, you can see some pink color. So
Yeah,
Oh, yeah, it's the spores are starting to
yeah.
mature. Mm.
cool.
Very.
Okay. Do I have more?
I think that was it.
Okay.
Thank you very much.
Thank you.
We got Dave and Doug.
I can go. I just have one. Chris, can you put the still image up first from iNet? Can
Oh,
everybody hear me?
Yeah.
Yeah, we were walking in woods above the Schuylkill River near Phoenixville, Pennsylvania, in Canal Park, and just looked over the dead log and saw these dark brownish black blobs. They were off trail about 30 feet, so hiked over there, and this is a slime mold. Now, I wasn't quite sure. I had seen a Stemonitis in the past, and I thought right away it's that chocolate brown chocolate tube slime mold. but looking at I think someone else, I think Jason showed us some. Are these called Capilicium? The thread-like spore-bearing structures, the sporangia. I think that's what they're called. And these are very long, and they have this roughened appearance if you look around in here. Whereas the sister genus Stemonaria has, which I think that's what this is. Sorry, the Stemonaria has the rough outer surface of each spore-bearing structure, these thread-like objects here. I mean, the plasmodium goes around, and then within a day, it'll make a rigid rod-like structure, climb up it, and then produce thousands of these spores. And apparently the Stemonitis, the chocolate tube, that's Stemonitis splendens, gives you a very smooth, just the way the structure is made. These are called nets that surround it. So if you see this rough, according to what I could find, it's more likely Stemonaria than Stemonitis. They're both pretty widely distributed fungi. And I thought this species was consistent with longa. Maybe someone else could say something. Tried to take a video, Chris. Maybe you could show that video. It doesn't last long. We had to clip it because it was too many megabytes. Do you have that? A second email? Yeah. It's going to go by very fast. It's a little dark, but you can see we touched it with a stick, and all these millions of little spores just popped right out, and was kind of neat. You can't see it too well in this video. It's a little dark, but these things are just loaded with millions of spores, and they don't really, like all slime molds, the plasmodia just moves around and eats up bacteria and small protozoa and things. It's a one cell deal, and it doesn't really penetrate the wood. It's not causing any decay itself. It's just sitting on the surface, and it's pretty harmless. And but the threads are interesting, and just wondered if anybody had seen this, and if you think I'm right with the Stemonaria versus Stemonitis. There's several, apparently 14 species in the Stemonaria genus.
I think if Brandon is still here, maybe he knows. This is a fairly large clump.
Yeah, I mean, these threads are long. I mean, they're 3/4 of an inch. The clump was large.
Yeah, these things they require a lot of work, and you have to find the perfect fruiting body. So personally, I would just leave this at Stemonitaceae. But yeah, the ID that you put on it is not out of the question, I think. But yeah, I think you have to find the perfect fruiting body. Were these wet at all? Like, had it rained? You're muted, Doug.
Can you hear me now?
Yeah.
Yeah, no, it hadn't rained in a couple of days. They were very dry. The woods were very dry.
Yeah.
So
Okay.
my first impression of this photo was that it looked a little bit like Lemonitis, but the fruit bodies are kind of lying around and stuck together and curled up and
Yeah.
sort of disorganized. And Stemonitis, they tend to just sort of stand straight up, erect, straight.
Well, in perfect conditions, if you
Yeah.
find the perfect specimen, yeah. But Yeah. I mean, how big would you say each one of these individual fruiting bodies was? Like tall, you
Uh,
know?
yeah, between a half an inch and three-quarters of an inch. If you're talking about the individual thread-like
Yeah.
spore-bearing
Yeah.
structure, yes, they were long. Um, what I
And
found on
did you,
the internet was that like what Dave said, the images and the description, the Stemonitis, they tend to be in smaller clumps. They stand up a little straighter, and the surface of the capillicium, the spore-bearing structure, is smooth, whereas the Stemonaria, apparently they're surrounded by a net, and the net in the Stemonaria just sort of decays and leaves these rougher looking, less defined thread-like structures, whereas from what I could find in the internet, Stemonitis is much smoother. It
well,
also looked like,
that's just because the net is, has tighter openings, right? But essentially, all the Steminataceae, as far as I know, I could be wrong on that, but Stemonitis for sure. Um, it is also a net, and depending on what level of maturity you're finding it, those
mhm.
spores are going to be coming out of it. So it can look, not smooth because it's a maturity and that the spores are kind of puffing out of it and not making it look as smooth as um, again, that perfect fruiting body that comes from the textbooks, you know, uh, they don't always look like that.
Have
And then you
you,
also,
yeah,
and then you also really want to see how big the foot is compared to where that net actually starts. I'm looking through this photo. I don't think I can see a great example of that. Um, but maybe you noticed like that foot was really small. It must be.
yeah, if you mean the uh, closer area to the wood, meaning the foot, it was pretty small, and then these long thread-like structures came out of it. Yeah. Have you looked at the microscopy of these spores? Is there any difference between Stemonitis and
Oh.
this, or
Um, the spores are, yes, they're. I'd be lying if I said I knew the difference between this genus and Stemonitis and all the rest of the Steminataceae. That family is, it's not small. Um, but the most important thing I believe is to look at the shape of the net. So if you're going to do microscopy on these, you want to, you need good tweezers and pick up an individual fruiting body and actually blow the spores out. You want to look at an empty sporocarp. So you want to blow all the spores out of it, lay that on a slide, and have a look at what that net actually looks like.
hmm. But anyway, it's kind of interesting. It looks sort of like a carpet, like a shaggy rug. It's sort of neat looking.
Really cool find,
Yeah.
Doug.
Cool find. And if your rug looks like this, throw it out.
Yeah. It's like a dirty mop.
Yeah. Nice. Thank you for the find and the video. That's fun stuff. You ready to go, Dave?
Well, does anybody else have anything?
Igor also had some stuff that I think he was going to share. If
Yeah,
he's
I
ready.
need to take a break. I'll be right back. Igor, are you there?
You know what? I got one to show.
Okay, I'll be right back.
So right just before the meeting, I found this in my brick walkway, and it was just coming up in between the bricks. The only part that was sticking out was where the sand really stops on the stipe. And it was like camouflage dirty.
That's pretty cool.
Yeah.
You said you found this in a crack in the sidewalk?
I have in my backyard I have like bricks that are like, there's a decent, whatever, probably almost half-inch space in between
Yeah,
all the bricks. That's just, sand in between, and this thing just pushed up,
cool.
and I grabbed it not expecting much, but the thing just pulled out of the hole just like that.
Yeah, I mean, is this something if Igor's not here and Dave's not here, we might have trouble talking about this, but is this something in that section, uh, excavatus? Is that what he told me? No. Whatever, that section of,
This isn't, can we see the cap again?
not section excavatus, it's section vaginata.
Right.
But there's
Okay.
a subsection.
Well,
which
there's a subsection called penetratrices, and then you get
maybe,
the penetratrix, and I think that might be what this is. One of those things. Look at the cap; it's pretty distinctly striate, even though it's just hard to see because it's got all this debris all over it. So the question is, when you see something like this, there's a lot of dirt on it, so it's a little bit hard to assess. So is it a vaginate or is it an amidella? That's the question that comes to mind. I think the stem's a little bit long and thin and not ornamented.
What about the sac?
Amidellas have sacs.
What about
That's
striations?
that's the problem. Is that vaginate and amidella each have a membranous sac. Well, not all the vaginate. That's a really big section, but there's quite a few of them that have a membranous saccate vulva and no ring.
What about the presence of? Oh, geez. I was just going to say, what about a veil? Well, no ring.
Nope.
No veil.
Yeah. Well, apparently, I don't know. There's an argument going on now that I was just sort of ran across on MO, where someone is saying that all amanitas have partial veils, except a lot of them they're just so fleeting that you never see anything. But amidellas will sometimes leave remnants on the upper stalk, looking like maybe it was from a partial veil. And I actually have a photo of an amidella that has fairly well-defined ring on the stalk. The cap's not really completely open, so maybe that ring is going to disintegrate. But there's really nothing on the upper stalk of this. It's really quite bare. I don't see any kind of deposits at all. Also, combined with the pretty distinct striations underneath all that dirt and soil that it pushed up from in between, came in between bricks or something. You said, Chris?
Yeah.
Yeah. It's probably one of these penetratrices things. Amanita section vaginate, subsection penetratrices, or series penetratrices. I'd say that's probably what it is.
And does that name refer to its ability to penetrate up through sidewalk?
Yes. Yeah. The one thing that's missing here is most of these things in penetratrices have a really large, hard umbo right in the middle of the cap, and you sort of can't miss it. So, I mean, but this could just be, a species of section vaginate that's not placed in subsection penetratrices. It might just be, I mean, some of the other vaginate that I find do seem to originate fairly deeply under the soil, maybe as much as 2 inches. Amanita rooseveltensis, or perhaps what's a look-alike to that species. Sometimes the stipe is buried pretty deeply in the soil. But it's hard to get a read on what this species might be, or even what, what section it should, or what, not what, I'm pretty sure this is vaginate, but what, beyond that, where does it go? It's a little hard to tell because of all the dirt on the cap, but no umbo. No umbo sort of points away from penetratrices, actually. I think they all have that.
Yeah, this really doesn't have any of that. But man, was it down there? I was not expecting to pull up near as much mushroom.
Yeah, that's very interesting. So in between rocks or bricks or something, just a little space between the
Yep.
Yeah, that's pretty interesting.
Nice job getting it out all in one.
Yeah, you got
All
it out
luck.
of there.
Yeah, what did you
100%.
say? You just yanked it with your hand, kind of up, and
Well,
the whole thing, it came
I
out.
I just gave it like a tiny little tug to see, because I was gonna just move the brick, and like if it was, and it was like
Oh,
it was almost loose in there. I just barely tugged on it, and it just
generally
pulled.
speaking,
I thought it would be broken, but it just wasn't.
yeah. Usually the penetratrices, you can't just like give a little tug and it comes out. They're usually buried pretty deeply, and you have to dig them out. And if you're not real careful, you'll break the stock.
Yeah, but what about in sand? I've yanked up stuff in sand and got way more than I even thought I was
Oh,
going
and
to get.
sand. Yeah, well, that might make a difference. You're right. Sand
Yeah,
might
that
make a difference. Yeah.
like it was funny because when I looked at it in the brick, because it had just started raining, and I'm like, I gotta grab this thing, otherwise I'm not gonna get it.
Yeah, and I'm not making a case for
No, it's a good question that
penetratrices.
might make
but it but like
for that.
the stipe like appeared to be loose in the hole, which is why I was like,
Well, yeah,
maybe it'll give.
yeah. I mean, theoretically, the cap, you know,
Pushed
well,
all the
probably
way
did.
through.
Well, I don't know. Penetrate that. I don't know. That's why I had excavatus in my head, because I just learned about these at the foray from Igor, uh, or that subsection, whatever it is. Um, and that's why it
I'm
was just
trying
all
to
in my
think
brain.
of where you got excavatus from. Does that apply to any sort of Amanita? What is excavatus? Where do I know that from?
Um,
Um,
there's another mushroom that goes by excavatus that does the same thing, right? It just pushes stuff out of the way, which penetraxis kind of works the same way, I guess.
yeah, it starts from deep underground, and it's got the hard umbo, uh, because that's like the battering ram, to push stuff out of the way
Right,
as it's
right.
coming up through the soil and the rocks and so forth. Excavate.
Uh, is
I'm,
that
I'm going to look
foamies?
on Michael
It's a
Quebec. I have to see now.
it's a foamies, right? Foamies excavatus.
Oh, Flummies. Oh, yeah, and it's got nothing to do with this sort of thing.
No. Well, no, it does.
Flummies
It does.
excavatus, right?
Pushing
Thanks for
through the side
saying
of the log.
looking.
No, it makes the wood easy to excavate for.
Nice.
No,
I think
the
it's a
Flummies excavatus is called that because the margin around the cap hangs down like almost a centimeter, and it creates the impression that someone like dug out
scooped it out.
scooped out where the hymenium is. Yeah, I think that's why it's called excavatus. Fummies
See,
excavatus, that is.
well, this is also, that's not a common term, but it is, it's alluding to something. And I get this, it happens often because that mushroom is used by a woodpecker to pretty much hollow out trees.
Oh, okay. Well, maybe that's the reason. Could be.
I could be wrong on that. I'll look it up.
You find something, you share it.
Yeah, I
Yeah,
think for now you just want to say Amanita section vaginate.
that's all I really had.
Yeah. Pretty cool.
Yeah, it was just, just walking in between the house and the garage. And
Is Igor there? I guess we lost Igor because of the storm, maybe.
yeah, he said he might lose power.
Yeah.
I
heard him
was
say
gonna
that.
I was gonna pop in the chat. It got me. He and I live pretty close to each other. It got dark real quick.
These little things were in patches coming out of needle duff in a primarily Norway spruce stand. There's probably some hemlocks and pines mixed in. There's also some hardwoods mixed in, smaller beech or birch or whatever. So I thought this looked a little like Hydropus praedecurrens, but the gills are not decurrent enough for that species. I tried to get a spore print. I should have let it go longer because I had a lot of these, so I could have spared two of them. But I guess I had something to do. Sometimes I just seem to not have time to do all this stuff. So I wanted to examine the spores quickly, and there was not a real thick spore print. And I did I was able to scope the spores. I got a pretty good look at them, but I wasn't able to tell if the spores were strongly amyloid because there wasn't really a thick enough print to assess that well. But I think this mount here is in only Meltzer's. I should have written it down, and they're showing up pretty well. So I would say that, and the spore print was white. It was I could see that at least, even though it was very thin. So I don't know, maybe some sort of hydropus. So I've got here on MO, and this is one thing I like about MO. You can propose something like Mycena sensu lato, which is Mycena in the broad sense, which basically takes into account other genera that are related to Mycena that probably were split off genera, which I believe Hydropus is one of them. There were also some ones of these that I think were the same species that were white with a little bit of gray on, toward the disc. So I made a collection of these. I made a collection of the white ones. I preserved each collection separately. And these gray ones I sampled for sequencing. So maybe we'll find out. There were quite a few of them in these little patches in this primarily Norway spruce stand in the Poconos.
Cool.
Yeah, I know. What could you say about those things?
I feel like I've seen them before, but
yeah, me too. They look a little bit like Hydropus to me, and I believe I found Hydropus praedecurrens a couple of times. No, so this coral was in the same habitat. It was under the spruce. It looks to me like these corals are fruiting from litter, from like a lot of buried needles and stuff like that, more or less forest litter. The spore print was kind of pale, although the spores did show up mounted in KOH, so they're not like high line or very very hard to see. Even now, the KOH may have reacted with something in the spores and maybe darkened them a little bit. But I think this is Phaeoclavulina, it's not Ramaria. Phaeoclavulina are saprobic. Ramarias are all mycorrhizal, I'm pretty sure. The spores are pretty small. Ramaria spores tend to be a little bit more elongated and cylindrical, maybe. A little more like cylindrical. These are kind of like elongated pip shape or elongated sublacrimoid, which means shaped like a tear. So I don't know. I sampled them. I sampled them for sequencing, so maybe I'll find out. But I kind of like this proposal, Phaeoclavulina. There's a few species, and they kind of all look like this.
Isn't Like one of the things, like the Romarias with iron salts turn their color,
Yeah,
but
I
then
know. I was in a
the
hurry.
Clavulinas don't.
I was in a hurry. Um, iron salts is something to put on these corals because there are some distinctive color changes in at least some of the Romaria. But I think there's a color change on this Phaeoclavulina also. I'm not sure. Or maybe it's just one of them just stains green. I bring on so many mushrooms, I don't have enough time to really research all of them. Because I've been bringing home a lot of stuff just to try to sample for maybe for DNA, and maybe I'll do a little bit of microscopy. But a lot of stuff has just been going right into the dryer. I sample it first because so I have these, plates with wells to fill, and it's easier to do with fresh material, and it's probably better to do with fresh material. Also, I'm not sure, but I would think so. But anyway, yeah, kind of a cool little coral. This is in good condition. And Pluteus. So how do I know it's section Pluteus? Well, those are usually the bigger ones. This one's not very big though. It's kind of frail, kind of graceful. The flesh is not real thick. So I did some microscopy, and I found horned hymeneal cystidia with some of them with thick walls. This one looks like it has pretty thick walls. Oh, fairly. Oh, yeah, you can see up towards the apex. You can see where the walls are thick. It's easier to see. So that's a metuloid. It's a horned Cystidia. Then there's this other cystidia that these don't look like they're horned, but they do look like fairly thick walls. And I found a lot of cystidia. You can usually find a lot of cystidia on these section Pluteus. These particular cystidia are really long and thin, so maybe that matters. I'm not sure. Of course, I sampled it. Will we get a sequence from it? I don't know. If we do, though, the genus Pluteus is really interesting. There's a lot of species that just don't get mentioned very often because they're hard to determine, even with a microscope. So here's another thing that points towards section Pluteus. The horned cystidia were one thing. The pleural cystidia, the metuloids were the one thing. The other thing is this is the Pileopellis. It's a cutis. So it's basically these more or less parallel hyphae that make up the cuticle, the upper layer of the cuticle, I guess, of the mushroom. So I just peeled a little bit of it and cut up a little tiny piece and smashed it with some Congo red and a little bit of water because the Congo red can saturate the picture a little bit too much. So, and then there are these terminal elements coming off of the Pileopellis, and they have sort of rounded apices. So I don't know, if I was to research this, maybe I could make some sense out of those traits. But the point is that I've recorded them. They're traits that I recorded. I mean, if I do find out what species this is, then maybe we get a little bit of extra information about it. Other than just knowing like a DNA sequence in one picture, we have some extra information. And I believe this was fruiting from a decaying hemlock log. So that's also kind of interesting that it was fruiting on conifer. I think more often Pluteus grows on hardwood. So it was nice. Practice my microscopy on Pluteus.
I'm glad you got it sequenced because within section Pluteus, that microscopy can not be too helpful.
No, it's not. It's really not. I mean, so many
It's
of
cool.
them.
It is cool.
Have the same. Yeah,
like
it's nice.
It
It's fun to see, and they're not. The cystidia are not hard to find. They're easy to photograph. it's not like looking for like Mycena cystidia.
Right. Yeah.
Yeah. So,
And I was just going to ask, Cervinus group section Pluteus. It's not uncommon to have some fibrils on the stipe. It looks like this one has some action on the stipe as well.
I didn't really assess very well. Yeah, it does. It does. You can see it. You're right.
Yeah.
You can see it in the
Yeah.
photo. Actually, you can see it in the photo. Yes. It's got a
Cool.
little bit of flocose fibrillose kind of ornamentation on the stipe. Yeah. But once again, how many of them have that? I don't know.
Right. Yeah, exactly. But your point, if this was indeed on hemlock, pretty interesting there too. So
Yeah, I think that might point away from Cervinus. And also the stipe is kind of thin and it's kind of small and thin for Cervinus group. Well, Cervinus group, basically, a lot of people just use Cervinus group to refer to pretty much virtually all of the species in section Pluteus, which are sort of the bigger. Well, not actually, some of them look a lot different.
Well, yeah, I guess it's Pluteus group it's a clade of, right?
Yeah, I guess they're all related.
Yeah.
Genetically, I'm not sure.
But online and all that, any big brownish Pluteus pretty much gets called Cervinus.
Yeah, if I put this online and called it Cervinus group, probably nobody would argue. But I don't think it is, though. I think it's something else. I think it's in section Pluteus, but I think it's going to be one of these names you don't see very often, But I like to try to get a lot of Pluteus sequence because you get a lot of different names back.
Yeah, cool.
So it's not every day I bring home an anoscopy and I'm able to identify it to species like within a few hours. So first of all, just the look of this cap is pretty distinctive.
I mean, that's Rimos.
Yeah, it's really Rimos and it's shiny and it's Rimos, but it's breaking apart, but it's sort of breaking apart in a way that's at the same time kind of holding together. So why isn't it then? What is it? Pseudosperma I think it's Pseudosperma ramosa or ramosum. Why isn't it that? It looks like that. Well, that's the answer is coming. It's got a little bulb too. Some of the Anacebaceae grow out of a little bulb. This one had a little bulb.
And those striations go all the way down to the foot. That's pretty interesting on the stipe.
Yeah, there's a lot of interesting features to this mushroom. It was only one mushroom. I think I plucked this off a lawn actually. I believe I got this off a lawn, bordered by oak woods on one part and hemlock on the other. But here's the kicker right up. If you look at the spores, Asterospora. It's the name of the species. And if those spores are really strongly stellate, star-like, they have big nodes, big rounded, smooth rounded nodes. So when I saw that, I knew it was not pseudosperma. Those do not have nodular spores. And not true anoscopies are the only ones with nodular spores. And true anoscopies are the only ones that have these thick-walled cystidia. This one doesn't have real thick walls, this particular cystidium. Let's see, here's more.
That cystidia that we were just looking at, I like to think that wall, and you can see it here too. You can see the thickness of that wall way at the apex.
Yeah, near the apex, you can really see it on this one. You can see how that wall is really thick because the wall is so thick and almost up here where the neck is thin, the wall is comprising most of what this thing is. So you can kind of see that there. So yeah, this one's got a pretty thick wall. You can see that pretty well. And it looks like the apex is ornamented, although sometimes it's hard to tell because it might just be a spore that's stuck to the apex. But I think that's an ornamented apex. So if you look on Mycoclebeckia, you'll see Inocybe asterospora. It looks just like this thing. Okay, the spores are right on target. And of course you always get, sometimes a variety of shapes, for the hymeneal cystidia. So yeah, but I'm pretty sure I've got this one identified to species, which, like I say, it's usually I'm not able to identify Inocybe.
Yeah, and I agree. I've only ever found it once. Can we cycle through? Was there more? Oh no, we got through all the microscopy. Cool. Yeah, it's a cool one.
Yeah, I thought so. Yeah, there were only one of them. A lot of times, if there's only one, I just leave it. But I thought this looks pretty cool. I'm going to take it home. And I managed to get spores out of a little piece of cap. Out of the same little piece of cap, I got a sample for DNA, using a toothpick. And so I got a lot of mileage out of this one fruit body, and most of it is now preserved. It's just like maybe 1/4 of the cap was cut off.
What did you use a toothpick for?
Okay, the way you sample for DNA for nanopore using these plates with wells is each well, there's 96 wells in a plate, and they have these snap-on lids. So you snap off the lid, and if you have fresh material, you don't necessarily need to dip your toothpick in the chemical solution first. If you're doing dried material, you probably should dip it in the chemical solution first, because that'll help get some spores to rub off onto the toothpick. So with fresh material, you just take your toothpick, rub it on the hymenium of the mushroom, twist it around a little bit so you're getting stuff onto the toothpick. You probably want to try to avoid getting actual tissue on the toothpick. So if you get like a chunk of gill, you probably want to try to get that to drop off, because I think what happens is I know Steve Russell told me, don't put actual fruit bodies or pieces of fruit bodies in the wells. I think what happens is probably they start to deteriorate. They start to decay. Because there's no special storage or anything for these plates. They're just sitting here in a room in my house. But you take your toothpick, the one end of the toothpick you rub on the hymenium of the specimen, and then you put that tip of that toothpick back into the chemical solution that's in the well in the plate. Swish it around for 2 or 3 seconds. Swish it around real good. Sort of move the toothpick around, So, and then apparently there's enough chemistry comes off of that toothpick that they're able to sequence the mushroom, based on what was on that toothpick that got dipped into the solution. So I'm waiting for feedback on what I've been doing. I've already sent like six plates.
6 plates at 96 each?
Yeah. Yeah, it's like having a full-time job. It's actually it's a lot of work, actually. But Steve is allowing me to send as much stuff as I want, partly because our little group here in Pennsylvania is funded with a $4,000 grant that Samantha got for us. But that $4,000 apparently can go a real long way when somebody in the group is willing to do a lot of the labor, which is basically what I've been doing. So, all of this sampling and then recording INET numbers and MO numbers on a spreadsheet. And I'm also drying the material as well, because I'll send that stuff to Samantha. And she's going to try to get as much as possible accessioned into the Philadelphia Museum of Natural History, where she works in the accessions department. So anyway, here's another mushroom. I'm pretty sure we've got here Lactarius subplymphogallis. Okay, try saying that 5 times fast. So this is interesting, though, because
Yeah.
I have another observation paired with this one that we'll look at in a minute. That was a question?
No, I'm just excited because I showed this and I didn't have that red staining, but you do. I see.
Yes.
Yes.
Yes. Mm-hmm.
Cool.
This one in situ, I picked them and the pink stain was already on them. But you know what I found out here, Hunter, is it takes a while for that pink stain to develop, because I dragged a knife across this one, the gills, and it took a while before I saw any pink. I'm not even sure I have a photo of the cut turning pink. But I also found another. No, I didn't. I didn't record. I didn't get spores from these either. I was too busy. I had too much other stuff to do. They just went right into the dryer. So these are white. Now, if you go to the next observation, it's paired up with this one. So it's sort of like one observation in a way. I think these are the same species. Look at the caps are gray. I'm pretty sure this is the same species. Now, are these like varieties of a species? Is there any genetic variation here? I don't know. I sampled both collections. I sampled the white ones, I sampled the gray ones, and these did not have pink stain on them when I found them. You see no pink stain at all here on these widely spaced gills. And of course, that's a key feature with this species. If I'm right, if that's what it is the widely spaced gills. There's white latex, and it took a long time for this latex to dry and leave behind a little bit. See how it's getting a little pink? This was after quite a while. I don't know if I wrote down how long this took, but it was a while. It was at least a half an hour, let's say. Maybe I didn't say. I should have said.
Very slowly.
Yeah, you know what? Though I can actually, I'm going to find out because I want to see now, because I could just look at the time signatures on my photos. So I'm actually going to do that right now because I can do this and not sort of screw up what else I'm doing. So pictures. What's the date on that thing? Is it like the
723?
Okay, so it's in this folder. And let's see. So we've got Lactarius subplymphogallis. All right, here's the gray one. Okay, I made the slice at 4:47, and I started to see the stain at 5:10. So almost a half an hour.
Oh, wow.
Yeah, almost a half an hour. And it was really the stain was not very dark at that point. But you could see that there was pink developing. And these are acrid. Both collections were acrid. The one collection was a little more quickly acrid than the other. The gray ones were sort of more immediately acrid. Are they the same species? I don't know. Let's hopefully maybe we'll find out.
That'll be good to see. Yeah.
I think that was it, Dave.
I think that's all I have. Yep.
I gotta get going, but I wanted to mention, did anyone see that Luke ate chlorophyllomylibidites? He said he claims the toxin can be cooked out, and he did it and ate it. But I'm not.
Some people eat that species and don't get sick. Is it a matter of cooking out the toxin? It is a matter of there being people existing on a continuum of reaction to the toxin. Is it a matter of cooking out enough of the toxin, if in fact you're able to cook it out? Or are some people just simply tolerant to it?
Yeah.
I should look in McIlvaine's book.
Oh yeah, and see if he.
See if he ate it.
Well,
Yeah,
he would have, right? Because
I
Yeah,
would.
he ate everything. Yeah, we'll see if he got sick.
yeah.
That's the thing. See if he got sick.
So I saw Luke. Maybe I told you guys. I saw him do a presentation on Charles McIlvaine, maybe last month,
Oh
and
wow.
I didn't know that. One of the things that I found funny about that is his nickname was it Old Rot Gut?
Iron stomach or rot gut or something
Yeah,
like that.
old Iron Gut, maybe.
Iron gut, maybe. Yeah, something like that. Yeah, well, McIlvaine in his book describes Clitocybe illudens, which was the name back then for Omphallotus illudens, the eastern jack-o'-lantern mushroom. I probably said this already, but it just never stops being funny, really. He McIlvaine states in the this 1890s sort of elaborate style of English. The plants were of good substance, toothsome and flavorful, but others were sickened.
Yeah,
So you had like this potluck party, everybody got
I
sick.
liked them. It's a shame that everybody else had a horrible time.
Yeah. What's the matter with you
this,
people? These are good.
And this is coming from the guy who has eaten, possibly hundreds of other allegedly poisonous mushrooms. I think the number of mushrooms that he ate, if I'm not mistaken, was over 1,000 species.
Probably.
Wow.
Yeah, I think so. Well, he also described Clitopilus delabata, which has some other sodorifica or something it's called now. I don't know. But it's called the sweating mushroom. We've had 2 incidents in our local club where people ate these things. They found them on their lawn, and one ended up in the emergency room because she didn't know what was happening to her. The other one figured out what she ate and literally sweated it out. Because these things make you sweat. They make you sick. You get a temperature. It's like you have the flu, and they're known to have been fatal. And what does McIlvaine say about them? Exceedingly like ivory. And then he says like something about them tasting good or something. I don't know, but it wouldn't surprise me if McIlvaine misidentified them and ate something else.
Is that talk available? Do you know where that was?
Yeah, it was at the Philadelphia Natural History Museum.
Okay.
Yeah, oh no, not the Natural History Museum. Academy of Sciences.
Oh, okay. Cool.
Yeah.
Cool. Thanks.
And he gave it to the Philadelphia Mycology Club, which he lives in Pennsylvania, and McIlvaine was from Philadelphia. So,
yeah, Luke did his fundus project on the mushrooms, the Philadelphia mushrooms of McIlvaine or something like that. He tried to find species that maybe were not like right in downtown Philadelphia, but were found maybe in that county or some regional respect that McIlvaine had described. So
right. Yeah. He knows a lot about McIlvaine. He is inspired by McIlvaine. And if you know Luke, hearing Luke talk about something that he's inspired by makes you inspired by it, right?
Right.
Luke's a good dude.
Yeah, he seems like it from the old Taxonomy Tuesdays. I wish he was still here. That'd be nice.
Yeah. Our club brings him up here once a year, like in early April, to do a cooking demonstration in the restaurant where we meet.
Nice.
And he gives very nice presentations, and the people love it. Cool. And it's one of our most popular events. Like the place gets jammed, when he's going to give one of these cooking demonstrations.
Fun. Sounds good.
He gave me some advice when he started looking into McIlvaine because, where he was finding fungi, and especially rare things that hadn't been, that were described only by McIlvaine, right? Where I live in New Jersey, there's this guy, Ellis, who around the same, actually it was about 50 years, maybe later than that. But that I come across species names that only Ellis has ever described. So he gave me some good information about, how to look into Ellis's personal life. So I was inspired to kind of do the same thing with Ellis here in South Jersey. So if anyone wants to help out with that, let me know.
Hey, it's a little research project. All right, guys.
I love the mycological history. I love it. All right.
And McIlvaine worked closely with Peck and Mary Banning. So if anyone has seen the Mary Banning presentation, I believe McIlvaine is included there.
Yeah, I've heard a little bit about him. And yes, I saw that. Yes. Cool. Well, I'm out of here. Thanks, everyone. See you next time.